So hello everyone, thank you for assisting to this presentation. So today I will present my research project which is about the health beneficial bioactivities of Canadian faba bean. So as you know, currently there is a growing demand for high quality and more sustainable protein sources. And in order to fulfill this demand, faba bean is a very interesting alternative because it contains a very high protein content. Also, it's a non-GMO, gluten-free, and not a regulated allergen, which are all characteristics that can be helpful in the food industry. However, faba bean is still underutilized, so its health-promoting properties are not well understood and well studied if compared to other widely used legumes. So this is why we have selected to investigate these properties through this project. So this is an overview of the methodology that was used. So in this project, three novel Canadian varieties have been compared. And they have been compared to two control legumes, which were pea and soy, because they are widely used in the food industry. So those legumes have been processed, and then they have been digested in vitro using the standardized INFOGEST protocol. And after digestion, the small and highly active molecules have been enriched by ultrafiltration. And of course, these molecules included bioactive peptide. And then in the digested permeate, three bioactive properties have been investigated. So the antioxidant, antihypertensive, and anti-diabetic activities, and those were selected based on the result of an in silico analysis. I will discuss some of the main, most important results of my research. So first, if we talk about the antioxidant activity, as you can see, several assays have been used in combination in order to target various mechanisms of action of antioxidant. So both in vitro chemical assays have been used, as well as a cellular model to assess this property in a more physiological relevant model. So for the chemical assays, there were no significant difference among the faba bean varieties, except for the ABTS assays, as you can see here. And overall, the antioxidant activity of faba bean was either similar or better than peas, or similar or lower than soy. For the cellular model, the outcome is slightly different. So as you can see, for the Fabelle and Snowbird variety, the antioxidant activity was superior to pea and soy. So this is a very interesting finding. And this could be explained by the fact that the peptides that are generated by faba bean may be more resistant to intestinal brush border, or they can as well have a better bioavailability. So of course, these elements will have to be further investigated. For the antihypertensive activity, the faba bean had the Fabelle and Malik variety had a better activity compared to pea, but a lower activity than soy. And for the anti-diabetic activity, all the legumes investigated had an interesting activity. However, there were no significant differences among samples. So to conclude my presentation, as you can see, faba bean has a very interesting bioactive potential, especially for antioxidant and antihypertensive property, and also especially if compared to peas. So eventually, faba bean could be valued as a health-promoting food ingredient, but of course, further evaluation of this potential in vivo will be required. So thank you for listening to my presentation, and I will be happy to answer some questions.
Thank you, Delphine. I neglected to introduce Delphine. This is Delphine Martineau-Côté at McGill University in Canada, and she has spoken to us about faba bean flour. So there are some questions. I was very surprised about the differences in your cellular antioxidant activities. They were striking, especially based on the peptide size. Are there many more different types of faba beans that would be tested, or I mean, you tested 5. How many are?
I tested 3 faba bean varieties,
okay,
and 1 soy and 1 pea varieties just to compare.
Okay. That's right. I got confused there. So what platform did you use for your in silico predictions?
I used the Biopep database. So I used the proteins sequence of globulins of faba bean to verify if they were a bioactive fragment among this protein when they were digested with trypsin, pepsin, and chymotrypsin.
Thank you. Would any of our judges like to pose a question?
Yeah, I maybe can start. Thank you, Delphine. Very nice presentation. Very interesting because it's opening a lot of questions, not only on faba bean, but also on other pulses. Which protein, which type of protein extraction did you apply before going to this digestibility? Because you are speaking about globulin. Is it the full protein package, or is it just one fraction?
The globulin was just for the in silico analysis, but for my research for now, I just use a flour because we wanted to have the whole matrix in order to be more representative on how the faba bean will be consumed. But eventually, of course, it would be interesting to compare with purified protein to see if the same activities can be obtained.
Okay, great. And do you know if the faba bean varieties you were using were low in visin and convisin, you know, because in terms of digestibility, you have the favism, and you need to pay attention to this because it can interact maybe with your experiment.
Yeah, of course. So the 3 varieties that I've studied were different. So the Fabelle variety is low in visin and convisin. So this is an important element. The Snowbird variety was low in tannin. And the Malik variety is high in both.
Okay, thank you.
Okay, probably I can go next. Your poster looks really great. Congratulations, great job. I have one question about your experimental design. Because your sample, the 3K permit, is mixture. It not only contains bioactive peptides, also include like polyphenols, right? So my question is why you use the in silico approach to screen the bioactivity only based on the bioactive peptides?
Yeah, so my hypothesis is that most of the bioactivity that I've observed are associated to bioactive peptide, but of course I have a complex mixture. So polyphenol and also small oligosaccharide can have an effect too in this aspect. But for my further research, I want to purify further my permeate and by size exclusion, and also want to identify the peptide present by MS to confirm that those effects are really attributed to a bioactive peptide.
Great, thank you.
Thank you very much, Delphine, for your nice presentation. I think I have 10 questions. I don't know if I have time to ask all of them. But I like to ask you the first question. When you did the digestion, okay, I assume that you're using acidic digestion. Did you also add the amylase in your digestion?
Yes, I used the INFOGEST protocol. So I used the standard method. So I've used also amylase because there was starch in my extract, in my flour.
Okay, that's very good. So after digestion, so you have this digester, right? So what is the the protein content in your digest?
In my digest, in my permeate, or?
Yeah, I mean, after you finish your digestion, so you have digester, right? So
yeah.
what was the protein content in the digest?
Yeah, I've measured the protein, well, because my primary method is in liquid form, so I've measured protein with the BCA assays. If I remember well, it was around 5,000 microgram per ml of protein.
Okay, let me ask the last question. Do you have a plan to measure the bioavailability of the peptides?
Yeah, this is my next step in my project. So I want to see if those peptides can cross the intestinal barrier using Charcot-2 cells. So just to see if those peptides can really have an impact in vivo after that.
Thank you.